Hello everyone, a very warm welcome to the official page of Read My Course. I am Dr. Shatabdi and today we will be continuing with part 3 of the cell, the basic unit of life. So what are we going to cover right now? We will look at the characters or the features which are exhibited by an eukaryotic cell. Then we will look at some comparisons between a plant cell and an animal cell. Further, we will move and look into the structures which are present within a typical eukaryotic cell, starting off with the cell membrane, the cell wall, the endomembrane system, plastids, mitochondria, etc. Moving on to the next slide. So what are the unique features of an eukaryotic cell? The first most important feature is the presence of membrane bound organelles. Okay, this is totally absent in prokaryotes. We have already discussed this. Next is the presence of an organized nucleus, which is present within a nuclear envelope. This is also absent in prokaryotic cells. And the third important structure is that the genetic material is organized into chromosomes. So these make the eukaryotic cell different from a prokaryotic cell. Moving on to the next slide. What are the major differences that we notice in a plant cell and an animal cell? Some of these we have already discussed in our last class. For example, in a plant cell, we have a structure outermost covering which is known as the cell wall, this green colored structure over here. Another important thing that you are noticing is the presence of this large space. This is a central vacuole. You will notice that this is absent in an animal cell. Okay, and the third important thing that we have to look into details is the presence of a structure called chloroplast. These chloroplastids are basically helping in the photosynthesis of the plants, and this is absent in animal cells. Now coming to the animal cells, what is unique in the animal cells? In the animal cells, we find a structure which is known as centriole. The centrioles are absent in plant cells. So moving on to the next slide. What is a cell wall? So we have already studied that cells walls are present specifically in plants, in fungi, but not in animals. Okay, it is basically nothing but a non-living rigid structure, okay, which is present just as an outside covering of the plasma membrane in plant cells. Another important thing is that a cell wall known as the primary wall is present in case of young immature plants. As the plant gradually matures, this cell wall, the thickness of the primary cell wall diminishes and we find the presence of a secondary cell wall. In this picture over here, we can see that the green layered structure, outermost layer is the primary cell wall, which with the inner layer as the secondary cell wall. And another structure which we are noticing over here is the middle lamella. This middle lamella is nothing but it helps to glue together or stick together the neighboring plant cells. So what is the function of the middle lamella? The middle lamella sticks together or binds together the neighboring plant cells. With that, we move on to the next slide. Now we come to the plastids. Plastids are again present in plant cells and in euglenoids. And what is the specific function? The specific function depends on the color of the plastid. There are basically three different types of plastids. One is the chloroplast, then we have the chromoplasts, and then we have the leucoplasts. Okay, the chloroplasts basically contain chlorophyll pigment. They give the green color to the cells and mainly they are responsible for photosynthesis. The chromoplasts have carotenoids and they have xanthophylls. These are pigments which give the yellow, orange, green, these type of colors to the plant cells. So that is why you will find a red chili is red, a red capsicum is red. Why is it red? It is red because of the presence of carotenes. Okay. Next, moving on to the next slide, we have the cell membrane. The cell membrane is actually quite a complex structure, but I'll try to explain with this diagram over here. See, the cell membrane basically consists of a phospholipid layer. Okay, the phospholipid layer, along with that we have some proteins and we have some carbohydrates in that layer. Now the phospholipid layer is a bilayer. What do I mean when I say that it is a bilayer? The fact is that, as we can see in this picture, it has, this is one layer and this is the other layer. So it is a bilayer, phospholipid bilayer. So each such phospholipid has a head, which is the phosphate head, and a tail, which is made up of fatty acid. Okay. So the phosphate head is the polar head and the tail portion is the non-polar portion. That we have to remember. That is, the round heads are the polar heads and the tails are the non-polar heads. So next we move on to the proteins. There are two types of proteins in the cell membrane. One is the carrier proteins and the other is the integral proteins. Third one is the peripheral proteins. So integral proteins are basically proteins which are embedded within the channel, as we can see in the diagram, and peripheral proteins are proteins which are present extending outside from the cell membrane. Okay, so with that basic clear, we move on to the next slide. Now a very important concept again, the fluid mosaic model of the cell membrane. The fluid mosaic model of the cell membrane was given by Singer and Nicholson in 1972. Remember this, the fluid mosaic model is a model given for the cell membrane. It was given in 1972 by Singer and Nicholson. What were they saying? They said that the cell membrane could move in such a way that they give the name quasi-fluid to it. Quasi-fluid. What's the spelling? It's Q-U-A-S-I, quasi-fluid. That is, apparently it seems to have lateral movements in between it. Okay, that is the quasi-fluid. So because of the presence of this lipid, phospholipid bilayer, this was possible. And what is the main function? What is the main function of this fluid mosaic model of the cell membrane? Functions like cell growth, formation of the intercellular junctions, then secretion, endocytosis, cell divisions, these are all the functions. Moving on to the next slide. What is the most important function of a cell membrane? The most important function of a cell membrane is transport of molecules. I remember we had already studied in the last class that the cell membrane is selectively permeable in nature. Okay, it means that it allows substances to move in and move out across its channel, but in a selective way. Now I'll try to explain this. Basically what happens is we have four different types of movement which we notice in this cell membrane. First is passive transport, next is diffusion, third is osmosis, and the fourth one is active transport. Now just have a look at this picture over here. As we can see in the first part of the picture, the red molecules are in abundant outside the cell, whereas they are less inside the cell. Okay, so when a molecule moves from its area of high concentration to its area of low concentration, it does not require any energy. It is a simple process not requiring any kind of energy. Involvement of energy is not there. So this is known as the passive transport, from its higher gradient to its lower gradient, from its higher concentration to its lower concentration. Okay, that is passive transport. What is active transport? Look at the picture. We are noticing that inside the cell, we have a higher concentration of a particular molecule, but outside the cell, it is less. But still, the arrow is from the outside to the inside, that is from the lower concentration to the higher concentration. Now, whenever a substance tries to move from its lower concentration to its higher concentration, then we require a carrier protein to carry it across the membrane. Also, we require ATP, that is energy. So, whenever there is use of energy or there is use of a carrier protein, that is known as active transport. So, I think by now it's clear what is the difference between active transport and passive transport in a cell membrane. So, let's move to the next slide. Okay, the endomembrane system. This is a very important part of any eukaryotic cell. The endomembrane system consists of four parts, that is the Golgi apparatus, the endoplasmic reticulum, the lysosomes and the vacuoles. These four together constitute the endomembrane system. First, we will read about the endoplasmic reticulum. Now, the endoplasmic reticulum is attached to the outer surface of the nucleus. Okay. Now, these are in the structure of tubules. That is, they have a tubular structure extending into the cytoplasm from the outer end of the nucleus. Now, when ribosomes remain attached to the surface, to the outer surface of these endoplasmic reticulum, they are known as rough endoplasmic reticulum. You can see in this picture that small dots are attached to the outer surface of this endoplasmic reticulum. These are the ribosomes and therefore that is the rough endoplasmic reticulum. Similarly, when you look over here, you see that these endoplasmic reticulum have no attached ribosomes to their outer surface. That is the smooth endoplasmic reticulum. So, two types of endoplasmic reticulum: rough endoplasmic reticulum with ribosomes attached, smooth endoplasmic reticulum, no ribosomes attached. So, when their structures are different, it's quite obvious that their functions will be different. So, what is the function of rough endoplasmic reticulum? Just think, we have ribosomes attached. So, what is the function of ribosome? Protein synthesis. We have already read. So, rough endoplasmic reticulum is involved in protein synthesis and secretion of the protein. Okay. Coming to smooth endoplasmic reticulum, because ribosomes are not attached, so its function is synthesis of lipid. So, rough endoplasmic reticulum, protein synthesis, protein secretion; smooth endoplasmic reticulum, lipid synthesis. Moving on to the next slide, Golgi apparatus. Now, these Golgi apparatus are cisternes or sac-like structures. They are present in very close approximation. They are present in very close approximation to the nucleus, okay, just outside the nucleus, and they have lumen and cistern structures. Also, they have two phases. We will discuss these phases in the next slide, that is the cis phase and the trans phase. What is the main function of the Golgi apparatus? Function of packaging materials. So, if you try to remember that Golgi apparatus is basically the courier system of a cell. What it does is it packs the materials and it delivers the materials. Okay, so just remember in this way that Golgi apparatus is nothing but the courier system of the cell. Okay, specific functions: packaging materials to be delivered either into the intracellular targets or into the extracellular targets. So with that, we move on to the next slide. Yes, in this picture, just have a look at this picture. We are trying to show the cis phase and the trans phase. This is the cis phase, this is the trans phase. What happens is there are some transport vesicles which move from the cis phase towards the trans phase. And as they travel from the cis phase towards the trans phase, and when they are excreted, they form secretory vesicles. Okay, this is important. Why? Because the secretory vesicles further will help in the breakdown or degradation of food particles. So, what happens is Golgi apparatus, it has two phases. One is the cis phase, another is the trans phase. Next, moving on to the next slide, we have lysosomes. Lysosomes contain lytic enzymes, basically hydrolytic enzymes. If we speak specifically, they are hydrolases, they are lipases, they are proteases, and they are carbohydrases. So, with all these lytic enzymes present in between them, they are mainly responsible for digestion of carbohydrates, proteins, lipids, and nucleic acids. Just have a look at this picture. In this picture, I'll try to zoom it and show it to you. See, in this picture, food particles are entering from one side of the cell into vesicles. Okay, then they are becoming bound by vesicles. In the meantime, these dotted structures which are present encircled, they are the lysozymes. Those lysozymes, these are the lysozymes. These lysozymes get attached to the vesicles. Then what they do is they release their lytic enzymes, which cause degradation of the food particles which have been ingested. Ultimately, what happens is exocytosis and the release of the digested products. Is that clear? Okay. Next, moving on to the next slide. Vacuoles. Vacuoles are nothing but membrane-bound spaces which are found in the cytoplasm. They are found both in plant cells and in animal cells, but we must know that in plant cells, they make up almost 90% of the volume of the plant cells. We can see in this picture. As we can see in this picture, see, this is the central vacuole, okay? And look at how much space it is taking up inside a plant cell. So, we remember that it is taking up almost 90% of the space within a plant cell. So, a question comes up that what is tonoplast? See, tonoplast is the membrane or the covering of the central vacuole, that is known as tonoplast. Just remember that in amoeba, we have a contractile vacuole. This contractile vacuole helps in osmoregulation and excretion. So with that, we come to the end of this session. In the next session, we will study in details about much more important structures like the mitochondria, the nucleus, the centrioles, etc., which are very important structures of the eukaryotic cells. Till then, take care, and if you like this video, please do subscribe and comment in the comment section below. Thank you.